Okay, so welcome back to this next video in which we are discussing cyclic AMP signaling. Okay, right, so we're in the process of discussing the GS cascade. Okay, so we have seen that GS-coupled G-protein-coupled receptors, such as the beta-adrenoreceptors, when they bind their ligand, they can then activate GS heterotrimeric G proteins. Okay, so the off-state GS heterotrimeric G protein will bind to the intracellular aspect of the G protein-coupled receptor once it's in the on conformation, i.e. it's got its ligand bound, and that will then catalyze the release of the guanosine diphosphate from the alpha-S subunit of the GS heterotrimeric G protein, and it will be replaced by a GTP molecule from the cytoplasm. Okay, when the GTP binds to the alpha-S subunit, the alpha-S subunit changes conformation so that it no longer binds to the beta-gamma complex, and therefore the beta-gamma complex cleaves off the alpha-S subunit, okay, and it also cleaves off the G protein-coupled receptor, and it goes off, still attached to the underside of the phospholipid bilayer, and we're throwing that one away for now. We don't care about that for now. Okay, right, the bit we care about is the alpha-S subunit with GTP bound. That's now on its own, and it's also going to cleave away from the G protein-coupled receptor to which it bound, okay, and it is also going to go off on its own, attached to the underside of the plasma membrane by a lipid moiety. Okay, and these alpha-S subunits in the on state are capable of activating all 9 plasma membrane adenylyl cyclases. Okay, so they can bind to the C1a, C2a dimer of plasma membrane adenylyl cyclases and promote that dimer going into the on state. Okay, so where it's actually capable of catalyzing the conversion of ATP into cyclic AMP and pyrophosphate, or at least that's the leading theory of how you activate your adenylyl cyclase. Okay, right, so this now has overall led to the production of cyclic AMP by our adenylyl cyclase enzyme. What we now want to look at is what are the targets for that cyclic AMP? What does the cyclic AMP do? Okay, and we're going to start off by discussing the most famous target for cyclic AMP, and it's not the only target for cyclic AMP. Later on in the video we'll discuss the exchange protein directly activated by cyclic AMP, Epac, and we'll also discuss cyclic nucleotide gated ion channels. Okay, but the main target of cyclic AMP, the most famous one, is protein kinase A, the one that's received the most attention. Okay, so protein kinase A is the next topic we want to take on then. Okay, and protein kinase A has another name. It's also called the cyclic AMP dependent protein kinase. Okay, and this is the slightly more fancy name for protein kinase A. If you read the literature, generally reviews and research articles don't call protein kinase A, they call it cyclic AMP dependent protein kinase. Okay. Now, what other names can you use for it? Well, you can abbreviate protein kinase A down to pKa, so you often see it written like that. Okay, right, so protein kinase A is quite a complicated enzyme. Okay, it is not just one polypeptide, unfortunately. The inactive protein kinase A is actually a tetramer. Okay, so I want to discuss firstly the structure of protein kinase A with you, and then we'll discuss how protein kinase A enzymes can actually be activated. Okay, right, so as I say, the inactive protein kinase A is actually a tetramer, okay? So inactive protein kinase A is a tetramer, okay? And it's made up of 2 regulatory subunits that are dimerized together with 2 catalytic subunits bound to those 2 regulatory subunits. Okay, so it's what we could call a heterotetramer, because the different subunits that make up the protein kinase A are not the same sort of protein. Okay, so I'm going to start off by discussing the regulatory subunit dimer, and then we'll add on the catalytic subunits. I'm going to tell you how to build a regulatory subunit dimer of protein kinase A before we then complete the protein kinase A molecule by adding on catalytic subunits. Okay, so firstly then let's have a look at the regulatory subunit dimer. So basically, to make the regulatory subunit dimer, you're going to join together two regulatory subunits of protein kinase A. Now let me show you some of the broad structural features of the regulatory subunits of protein kinase A. So these are individual proteins. So here is the amino terminus. Okay, then a really important domain is going to be this first domain here at the amino terminus, which we're going to see in more detail later, but for now we'll just abbreviate it as a box. And this domain is what's known as the D/D domain. Okay, now what does that stand for? Okay, well the first D stands for dimerization. Okay, and the second D stands for docking. So this is the dimerization/docking domain. Okay, right, and this is actually going to be how the two regulatory subunits are going to dimerize together. So if I start by drawing my second regulatory subunit of protein kinase A here, here's its amino terminus, and here's its dimerization/docking domain, and they're going to be dimerized together here. So I'll draw them both in orange. So these are the D/D domains dimerized together of two separate regulatory subunits of protein kinase A. Okay, now let's continue on our first regulatory subunit of protein kinase A. So what you then have is a portion that's known as the linker. Okay, and then it curves around like so. And then you have the portion with the cyclic AMP binding sites in. Okay, and each regulatory subunit of protein kinase A has in total 2 binding sites for cyclic AMP here. Okay, so let's highlight up these different portions. So this portion in green here, this is the linker, and it's in this little crack here that we're actually going to bind the catalytic subunit in a moment. Okay, but I'll talk about that later on. Okay, so this green portion is known as the linker, and it's the linker between the dimerization docking domain and the cyclic nucleotide binding domains down here. Okay, so we'll then colour in this portion with the 2 cyclic AMP binding domains in blue here. So this is where the binding sites for cyclic AMP are. Okay, now there are 2 separate binding sites then for cyclic AMP on each regulatory subunit of protein kinase A, and these have special names. The one that's closest to where the catalytic subunit is going to bind is known as the cyclic nucleotide binding domain A. Okay, so CBD, I'll put this here, CBD stands for cyclic nucleotide, so that's the CN. Okay, and in fact some people will occasionally put an extra letter in this and will call it the CNBD, okay, so that you've got the nucleotide there, but more commonly you see it just written CBD. So C stands for cyclic nucleotide, and then the BD is for binding domain. Okay, so that first cyclic nucleotide binding domain of the regulatory subunit of protein kinase A is called the cyclic nucleotide binding domain A. Okay, and the second one here is then called the cyclic nucleotide binding domain B. Okay, like so. Okay, and then you've got the C terminus after these 2 binding domains for cyclic AMP. Okay, now to complete up the other regulatory subunit here, this will just be the mirror image. So here is this linker region again. Okay, and then here we have the portion for binding, cyclic AMP with the 2 cyclic AMP binding sites there. Okay, so I'll just colour this in as well. So the linker regions here in green. Okay, and then the portion containing the 2 cyclic nucleotide binding domains is here in blue. Okay, right. So basically, it's more complicated than this actually when you build a regulatory subunit dimer like this, because there is not just one regulatory subunit of protein kinase A. So these proteins that we have built this regulatory subunit dimer out of, they are known as the regulatory subunits of protein kinase A. However, there is not just one type of regulatory subunit of protein kinase A. Okay, it would be really nice and simple if there was just one regulatory subunit of protein kinase A. You just built 2 identical of the versions of these proteins, and dimerize them together, and then you've got your regulatory subunit dimer, but that's not the way it is, unfortunately. Instead, there are 4 genes which code for regulatory subunits of protein kinase A, and these are classified into 2 families. Okay, so there are the type 1 regulatory subunits of protein kinase A, which for short are written R1 like this. Okay, so this means type 1 regulatory subunit of protein kinase A, and there are 2 genes in this family. There's the R1 alpha, the type 1 regulatory subunit of protein kinase A alpha, and then there is the type 1 regulatory subunit of protein kinase A beta, R1 beta. Okay, right. Then there are also type 2 regulatory subunits of protein kinase A. Okay, and there are 2 of these as well. There is the type 2 regulatory subunit of protein kinase A alpha, and then there is also the type 2 regulatory subunit of protein kinase A beta. Okay, so this makes it rather more complicated, because now the question arises, well, how do you build a dimer? Can you mix together any one of these 4 different regulatory subunits of protein kinase A, basically? Okay, well, the answer is no. There are certain dimers that you can build. Okay, so you can build absolutely all of the homodimers. Okay, so what I mean by that is that you can use the identical regulatory subunit of protein kinase A in the 2 slots for all 4 of these. So for instance, you can make a regulatory subunit dimer out of 2 type 1 regulatory subunit alphas. Okay, so you put an R1 alpha here and R1 alpha here, that's an R1 alpha homodimer. Again, you can do that for R1 beta. You can put an R1 beta here and R1 beta here. That's an R1 beta homodimer. And the same for the R2 alpha and the R2 beta. So you can build all 4 homodimers of these 4 regulatory subunits of protein kinase A. Okay, however, there is one heterodimer that you can build. Okay, and that's basically a heterodimer between R1 alpha and R1 beta, and that's the only heterodimer that is allowed. Okay, so a heterodimer is one in which the 2 regulatory subunits of protein kinase A that you bind together are going to be different. Okay, and you can make this out of an R1 alpha and an R1 beta, and that's the only heterodimer you're actually allowed to make in nature. So for instance, you can't make an R1 alpha R2 beta heterodimer. You can't put an R1 alpha here and an R2 beta there. That's not allowed. The only heterodimer where the 2 regulatory subunits are different in the regulatory subunit dimer that you're allowed to build is one where you put an R1 alpha here and an R1 beta here. That's the only form of heterodimer for regulatory subunit dimers. So that in total gives us a total of 5 different regulatory subunit dimers for protein kinase A. You can produce all 4 homodimers and then that single heterodimer. Okay, right. Now, your regulatory subunit dimer then will be classified as either a type 1 regulatory subunit dimer or a type 2 regulatory dimer. Okay, so if you have built your regulatory subunit dimer out of type 1 regulatory subunits, so either the R1 alpha homodimer, the R1 beta homodimer, or the R1 alpha R1 beta heterodimer, that will be called a type 1 regulatory subunit of protein kinase A dimer. If, on the other hand, you have built your regulatory subunit dimer out of R2 subunits, type 2 regulatory subunits, either the R2 alpha homodimer or the R2 beta homodimer, then that will be called a type 2 regulatory subunit dimer. Okay, and that will become important in a moment. Okay, so let's now move on past the regulatory subunits and actually have a look at the catalytic subunits. So these regulatory subunits, these aren't actually capable of phosphorylating anything. Protein kinase A, what's it famous for doing? It's famous for phosphorylating serine and threonine residues on proteins. It's a very famous serine-threonine kinase. Okay, but these regulatory subunits of protein kinase A, forming this regulatory subunit dimer, they're not capable of phosphorylating serine or threonine residues. It's the catalytic subunits that are capable of phosphorylating the serine or threonine residues. Now draw the little catalytic subunits in here now. Okay, so I'm drawing the catalytic subunits in the typical way that you show enzymes with a little active site there. Okay, so I'll colour these in in vivid purple here. So these are the catalytic subunits of protein kinase A. And basically, in the inactive form of protein kinase A, what happens is you build one of these regulatory subunit dimers. There is no cyclic AMP at the moment bound in any of these cyclic nucleotide binding domains, okay? And the consequence of that is that the regulatory subunit dimer can bind onto two catalytic subunits of protein kinase A. Okay, so here's one catalytic subunit of protein kinase A, and there's another one over there. Okay, and basically you can see the way I've drawn this, I've tried to suggest that the active site is, concealed basically. It's bound to the regulatory subunit, and it's not going to be actually able to phosphorylate anything when it's bound to the regulatory subunit. So basically these catalytic subunits of protein kinase A, these are the proteins that can actually add phosphate groups onto serine and threonine residues in other proteins. These are the serine-threonine kinases here. But when they are bound to the regulatory subunits of protein kinase A, they're not capable of doing anything, okay? These are completely and utterly inactive. So here now, this is the full inactive protein kinase A tetramer. We have two regulatory subunits of protein kinase A, and these are sequestering off two catalytic subunits of protein kinase A, okay, and keeping them inactive, stopping them from actually being able to phosphorylate serine or threonine residues within proteins. Okay, now, for short then, the catalytic subunits of protein kinase A are abbreviated as the C subunits, okay? And there are three different types of catalytic subunits of protein kinase A, I'm afraid. So just like there was not just one regulatory subunit of protein kinase A, there is not just one catalytic subunit of protein kinase A. There are actually three different types of catalytic subunit of protein kinase A. The catalytic subunit alpha, the catalytic subunit beta, and the catalytic subunit gamma. Okay, now they all do the same thing. They all phosphorylate serine and threonine residues within proteins. Okay, so what you do if you're going to build a protein kinase A inactive tetramer is you build your regulatory subunit dimer, and we've discussed the 5 different regulatory subunit dimers that you can build. Okay, and then you bind 2 catalytic subunits to your regulatory subunit dimer, and these 2 catalytic subunits can be any of the catalytic subunit alpha, catalytic subunit beta, and catalytic subunit gamma. They're all coded for by different genes, but they all do the same thing, and they can all bind to these little patches of the regulatory subunits of protein kinase A and be inactivated by binding there. Okay, so before you have generated cyclic AMP then, this is the form that protein kinase is in, okay? You've got the regulatory subunits dimerized together, and they're holding these catalytic subunits bound to them and inhibiting them, stopping them from doing anything, okay? So if you want to activate the catalytic subunit of protein kinase A, what you need to do is get it released from this horrible regulatory subunit of protein kinase A that is stopping it from actually being able to do anything. Okay, so before we just discuss cyclic AMP binding and activation of the protein kinase A tetramer, I just want to discuss what the difference between type 1 protein kinase A and type 2 protein kinase A is. Okay, so people all the time talk about type 1 protein kinase A enzymes and type 2 protein kinase A enzymes. So what does this mean? Well, basically, this refers to what type of a regulatory subunit dimer you have used to make your protein kinase A enzyme. So remember I told you that there were these type 1 regulatory subunit dimers and the type 2 regulatory subunit dimers, and I'll just remind you in case you've forgotten that already. Okay, type 1 regulatory subunit dimers were built out of type 1 regulatory subunits. So you either had the type 1 alpha regulatory subunit homodimer, where you had an R1 alpha here and an R1 alpha here, or you had the R1 beta homodimer, where you had an R1 beta here and an R1 beta here, or you had the R1 alpha, R1 beta heterodimer. All of those are type 1 regulatory subunit dimers. Okay, if you have built a protein kinase A tetramer out of one of those type 1 regulatory subunit dimers, then that protein kinase A tetramer is known as a type 1 protein kinase A enzyme. Okay, Meanwhile, you can probably now guess what a type 2 protein kinase A enzyme is. Okay, a type 2 protein kinase A enzyme is a protein kinase A tetramer where the regulatory subunit dimer that you have used to build that protein kinase A tetramer is a type 2 regulatory subunit dimer. And remember, that means that the regulatory subunits that you've used to build that regulatory subunit dimer are both type 2 regulatory subunits. So it's either an R2 alpha homodimer or it's an R2 beta homodimer. Remember, there are no heterodimers of the R2 subunits. Okay, right, so that's what the difference between type 1 protein kinase A and type 2 protein kinase A is. Now, the functional significance of the difference between type 1 protein kinase A and type 2 protein kinase A will become much more apparent later when we come on to discuss A kinase anchoring proteins, ACAPs. But for now, let me tell you that type 1 protein kinase A enzymes, i.e. where the regulatory subunit dimer is a type 1 regulatory subunit dimer, those are generally soluble. Okay, generally they are free in the cytoplasm. Okay, whereas generally type 2 protein kinase A enzymes, where the regulatory subunit dimer is a type 2 regulatory subunit dimer, those are generally bound to membranes in some way. So either bound to the cell membrane or bound to the membrane of intracellular organelles such as the nuclear membrane or the mitochondrial membrane or the endoplasmic reticulum membrane or the Golgi apparatus membrane. Okay, and it's bound there by interactions usually with other proteins that are bound to those membranes, or potentially bound there by binding to other proteins that are bound to that membrane. Okay, and we'll come on to that more later on when we come on to discuss ACAPs. Okay, so there is a functional difference between the type 1 protein kinases and the type 2 protein kinases, and it's simply because the type 1 regulatory subunit dimers are not as capable of binding to things which are membrane bound as the type 2 regulatory subunit dimers. Okay, so now let's come on to the actual activation of protein kinase A by cyclic AMP. So how does cyclic AMP binding change the inactive protein kinase A into the active protein kinase A? So this is quite complicated, more complicated than you might think. Okay, so basically, when you produce lots of cyclic AMP, the obvious answer is that now the cyclic AMP is going to bind to these cyclic nucleotide binding domains of the regulatory subunits of protein kinase A and cause the release of the catalytic subunits of protein kinase A. However, it's slightly more subtle. Okay, so basically, in order to actually cause the release of the catalytic subunit of protein kinase A from the regulatory subunit of protein kinase A, you need to bind cyclic AMP to the cyclic nucleotide binding domain A. Okay, so this one here, this is the important one that you have to bind cyclic AMP to. Okay, if you bind cyclic AMP in the cyclic nucleotide binding domain A, then you'll get a conformational change here. Okay, that will cause the release of the catalytic subunit of protein kinase A, and the catalytic subunit is then free. It's uninhibited now, and it can go off and phosphorylate serine and threonine residues within proteins. Okay, however, initially, the cyclic nucleotide binding domain A is not actually available for cyclic AMP to bind. In fact, what has to happen is cyclic AMP firstly has to bind to the cyclic nucleotide binding domain B, cause a conformational change down here, which makes the cyclic nucleotide binding domain A available for binding cyclic AMP. Then a cyclic AMP molecule has to bind to the cyclic nucleotide binding domain A, and that causes the conformational change down here, which releases the catalytic subunit. Okay, so basically, it's cooperative binding. A cyclic AMP molecule binding to the cyclic nucleotide binding domain B makes it more likely then that a cyclic AMP molecule will be able to bind to cyclic nucleotide binding domain A, and then cyclic nucleotide binding domain A is going to cause the conformational change which releases the catalytic subunit of protein kinase A once it has cyclic AMP bound to it. Okay, so you do need cyclic AMP to bind to the cyclic nucleotide binding domain B and the cyclic nucleotide binding domain A, but it occurs in this fancy way where first you get it binding to cyclic nucleotide binding domain B, and that allows it to bind to cyclic nucleotide binding domain A. So we describe this as cooperative binding, okay, where cyclic nucleotide binding domain B allows the binding of cyclic AMP to cyclic nucleotide binding domain A. Okay, right. So then, the final thing I want to discuss now in full generality is once the catalytic subunit of protein kinase A has cleaved away from the regulatory subunit of protein kinase A, what does it do? Okay, well basically it is what is known as a serine-threonine kinase enzyme. Okay, and for short, serine-threonine kinase is abbreviated as SIR and then FR kinase. Okay, so let me just remind you of the structure of a serine and threonine residue. Okay, and we'll see how phosphate groups can be added on to serine and threonine residues. Okay, so we'll start with serine, which is usually abbreviated just down to SER. Okay, so I'll draw this as a residue within a protein. So here is the amino group. Okay, here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, and again I'll draw it as though it's bound to the amino group of the next amino acid along. Okay, so this is the residue structure of a general amino acid. Okay, now in the case of serine, the R group of the amino acid is a methylene group with an alcohol group coming off like so. Okay, now let me show you threonine. Threonine is almost identical to serine. Okay, all you have to do to turn serine into threonine is add on an additional methyl group. Okay, so here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, drawn as that's linked to the amino group of the next amino acid along, and then the R group of threonine is just like serine, a carbon with an alcohol group coming off it, but now, this time, it has an additional methyl group coming off that carbon, and then only one hydrogen coming off that carbon. Okay, so that's what the difference between serine residues and threonine residues is. Okay, now, basically this alcohol group of the serine and the threonine residue can have a phosphate group added onto it, okay, and that's what protein kinase A is capable of doing. It's capable of catalyzing the addition of phosphate groups onto the alcohol groups of serine and threonine residues within proteins. Okay, now there is a special sequence of amino acids that the serine and threonine need to be in order to actually be phosphorylated by protein kinase A. So you know, serine and threonine residues, they're all over the place in proteins. Do protein kinase A enzymes phosphorylate every single serine or threonine residue in the protein? Well, the answer is no. Firstly, the serine or threonine residue needs to actually be available on the outside of the protein to actually be phosphorylated. And what I mean by that is, if we have some protein here, some blob, okay, if the serine threonine residue is hidden right in the center of the protein, then the protein kinase A enzyme isn't going to actually gain access to that to be able to phosphorylate it. So firstly, the serine threonine residue needs to be on the outside of the protein, available for the catalytic subunit of protein kinase A to phosphorylate it. Okay. In addition, not all serine threonine residues that are available to be phosphorylated on the outside of the protein actually are going to be phosphorylated. In order to actually be phosphorylated, they need to occur in a certain sequence of 4 amino acids, okay, which tells the catalytic subunit of protein kinase A that it is allowed to phosphorylate this serine or threonine residue. And basically this sequence of amino acids is, I'll just write it out and now explain it. It's an arginine, lysine, and I'm doing this in single letter amino acid code for now. Then followed by another arginine or a lysine, then followed by anything, which are abbreviated as X, and then followed by serine, threonine, the single letter codes for which are S and T. Okay, so R then. R is the single letter amino acid code for arginine. Okay, it wasn't given the single letter code A because A went to alanine. Okay, so let me just remind you of the R group of arginine. So I won't draw the full amino acid structure out, I'll just draw the R group of this amino acid out. Okay, so arginine's R group then consists of 3 methylene groups coming off the alpha carbon. Okay, and because I don't want to have to draw 3 methylene groups, I've drawn 1 methylene group and then put a bracket around it and then subscripted it 3, which means repeat this 3 times. It just gets me out of having to draw it 3 times. Then you have an amino group here, followed by a carbon, which then has another amino group coming off it, and then a double bond to a nitrogen atom, which then has a hydrogen group coming off it. Okay, so this is the R group of arginine. Okay, now at this position number 1 of this sequence, you don't have to have an arginine. It can either be an arginine or it can be the amino acid K, and the amino acid K is for lysine. Okay, it couldn't have the single letter L because L went to leucine. Okay, now the R group of a lysine residue consists of 4 methylene groups, okay, like so, and then with an amino group on the end, like so. Okay, so that's a lysine. So in this first position you have either an arginine or a lysine, so make your choice, whichever one you want. Then in the second position it's the same, have an arginine or a lysine, so one of these or one of these. Okay, then in that third position it doesn't matter, have whatever you like basically. You can put whatever amino acid you like. X doesn't stand for any amino acid. It doesn't stand for any specific amino acid. It means you can put in any amino acid you like there. Okay, right. And then serine or threonine in this position here. Okay, so all those 8 different combinations there, because there are 2 options here, 2 options here, and 2 options here. Well, even more actually, because there's 20 options there. Okay, all of those possible combinations are sequences which tell the catalytic subunit of protein kinase A to phosphorylate that serine or threonine residue that's in that sequence. Okay, so it's serine or threonine residues in these sequences that are actually going to be phosphorylated by the catalytic subunit of protein kinase A. Okay, right, so we'll call it there for this video. In the next video I'll say a few words about the amplification that occurs within this pathway. Then what I'll talk about is how you can reverse the signal, okay, how you can actually turn this signal off once you've removed the extracellular ligand that was driving the signal in the first place. And then we'll go on to talk about some very notable examples of where this signaling pathway is used.